What we did, we took human stem cells that were derived years ago from donated early embryos, or what's called iPS cells, cells that are taken from the skin and turned back into a stem cell state. These are very established technologies. And what we do this time is we put them in a special media that takes them to really, really early stages, what we call naive. So we have to use these human naive stem cells. And then we put each aggregates of 120 cells. And again, in about 1% of the aggregates, we can see that cells start differentiating correctly, migrating and sorting themselves into the correct structure. And the farthest we could get is day 14 in human embryo development. The human embryo makes all its organs basically by week 5 of pregnancy. And we really don't know a lot. It's a black box of how organ formation happens. And it's almost impossible to study this period because the embryo is very small. There are justified ethical limitations to really access such material. Therefore, the goal is really, can we bypass and create some kind of alternative model from cells, from stem cells, and try to make a model for a human embryo. By making these models, and that are very similar, and we can start with stem cells that are engineered, labeled, knock out a gene, and so forth, so we can really know which hormones and which genes are important for which organ. And from that knowledge, we can try and go back and improve our differentiation protocol. And maybe actually in these embryo models, which is in the end some kind of a differentiation protocols, there might be some cells that are useful actually that you can take and transplant, or take them and expand them and transplant them, or take them and test drugs on them and so forth. So they could be also themselves as a source of cells. I don't think we can ever really make a real identical embryo, just because in the end, the uterus environment is really the most normal environment, and it's controllable. I want to emphasize that if you're talking about trying to make a whole baby pregnancy outside the uterus, that is just impossible, because the human embryo is very big. Pregnancy is 9 months. I mean, even in mouse, where pregnancy is very short and the embryo are very small, we can only do so far 10 and a half days. So, so that is not, it's not our goal, it's actually also impossible, and there are no concerns about that at all.